Kristen Holmes, and I'm from California, so I went to University of Southern California, and I got my bachelor's in biomedical engineering. After that, I actually worked in industry for a couple years down in San Diego in quality control, and then I moved to, I'm originally from Sacramento, so then I worked up in Davis in process development, and it was, lots of fun, but I really wanted to go back to research that I had done a little bit of in undergrad. So that's really when I transitioned, went back to get my master's from San Francisco State. And that was actually a really unique program where they actually, I didn't go in with the intention of thinking I would get my PhD, but the program really identified students and helped to really bridge and give that confidence in research to continue on. And so I was able to actually work in a lab at UCSF, and so I was actually doing cardiovascular research at the time. And then that led me to coming up to, or applying for PhDs, and then I was accepted to the University of Washington. And actually, as you referred to, the ARCS, getting that ARCS fellowship was very important in the sense in deciding to come up to University of Washington, because I felt it was nice to feel like there were already kind of people that believed in me and were in my corner and supported me. And so that was very meaningful to me. And then now that I've been at the University of Washington, so where I am in the program, I'm a third year right now. So I just completed my general exam in the fall, October. So it was, nice milestone, and so now I'm just really focused on experiments at this point. So I've done my classwork, and I was actually able to do an outreach TA-ship while I've been here at the University of Washington. It's this program called BioQuest, and it's run through the Center for Infectious Disease, and they basically bring like 12 high school students from around the state, and you teach them like lab skills. Like the first day we're teaching them like how to hold a pipette, and by the end of it they're actually like running experiments. And so it was a very wonderful experience to be a part of. And actually one of my students, I've kept in touch with him and actually brought him in the lab a few times at the Hutch. And he's now a freshman here at UW. So, they are high school seniors. So, so now he's at UW. So it's kind of nice to hopefully be able to keep him in the lab or bring him in the lab a little bit more. But now I'm actually in Alice Berger's lab at the Fred Hutch Cancer Research Center. We're in human biology. And the focus of the Berger lab is really to use...
What is that word? I'm not catching it. Something lab?
Berger.
Berger.
Yeah, that's just her last name.
Oh, okay.
Yeah.
Thank you. I didn't...
Yeah, and so the focus of the Berger lab is really to use high throughput platforms to identify the unique underlying mechanisms specific to specific types of cancer. So instead of thinking of all lung cancer patients the same, the idea is basically to stratify patients based on the underlying mutation, and then have patients given specific treatments for their specific mutation. And so there have already been developed a few different what we call targeted inhibitors, but the protein that I'm looking at is, well, obviously we don't have one, and it's called, it's deemed undruggable, and so I'm actually looking at that protein to see if we can identify some opportunities for targeted therapeutics. And so actually through that project, I've been able to forge a collaboration with another lab at the Hutch, which is the Stoddard lab in the Basic Science Division. And so he actually focuses on protein structure and protein engineering, and so I've been able to kind of bridge these two very different kind of scientific approaches together, and so I'm learning a lot and having a lot of fun for sure. And that's basically it. Okay, great. Any questions?
She's a scientist, I'm not. You said that it's great to be collaborating with two different approaches to the protein.
Mm-hmm. Yeah, so...
If you could try once again to have a non-scientific person understand what these two different approaches are.
Okay, so I guess maybe it just helps to tell a little bit more about my project.
Sure.
So basically we have so in the Virtual Lab with the high throughput platforms, we basically looked at,
See, wait, do
Oh,
you mind if when
sorry.
I don't understand
Oh.
the word, I got that's the professor's lab, which is, that's even different to me to call it
oh,
a professor's name, but I got that one. And then the next thing we have, you said,
yeah, so we basically tried to use, we looked at all the different proteins in the
Oh,
cell
okay.
at one time, and then introduced or overexpressed our construct, and then looked at all the changes in the proteins. So if any of the proteins went up or down.
what caused them to change?
So we would overexpress our cancer gene, basically, that we're looking at.
Overexpress. Okay, that's an interesting use of that word too, overexpress cancer, thank you. I mean, this is stuff that is fairly basic, but
Well, but it's also, you know, it's not necessarily something that we discuss over breakfast, you know?
Okay.
So overexpressing cancer genes doesn't usually come up when I'm having my coffee in the
Yeah,
morning.
so basically, yeah, what exactly does that mean?
I don't
Yes.
know.
So we basically, have our cells that already have
Mhm.
this gene expressed normally at normal healthy levels. So we basically just went in and increased the level of protein that is in the cell for our cancer gene that we're looking at. And then we look at look at all the proteins in the cell which have gone up and down because of that gene we introduced. If that
And
makes
so
sense.
if I were to guess what I think you're saying, when you overexpress the cancer gene, you're trying to, it's sort of like increasing the amount of a drug you might give a laboratory animal. in
Right,
order
a
to
similar
try to
way, we're
get
trying
a
to-
response. So
Right,
you're
yeah, we're trying to stimulate the cancer, basically, and
Exaggerate
then
the
see.
stimulation
Mhm.
in order to
Coming-
be able to see
coming
what
from a cancer
proteins
cell.
respond to.
Right.
So, I thought protein is things I ate for meals. But these proteins are ones that can, perhaps have a, targeted therapy.
Mhm. Mhm.
She said
Because the
it's-
protein gives it something to grab on?
So yeah, the protein is. I probably should have.
Oh, no worries.
Some
It's-
slides or
No,
This is
no,
great,
it's okay.
great.
You're being very patient and good, this is what we
Yeah.
need.
No, so the protein actually is like, you could think of it, actually call it a switch, and so it's actually on and off. Normally, it turns on and off. However, like in the cancer context, it just basically is always on, like the lights are always on, and so that's when you get the problem with cancer, or that leads to cancer. And so basically that's why we tried to overexpress, like introduce, to simulate the cancer by like introducing our cancer protein, if you will.
And so
Okay.
you're looking for different proteins that
Mhm.
will, I mean, and the differentiating thing I've heard about Jim Olson and his work, and I've been aware of that, but this is differentiating proteins between patients?
Sort of, yeah, sort of, yes. I think,
What makes this new is what I'm trying to understand.
so, what makes it new is not necessarily the approach, but we've actually kind of found, found some really interesting data, actually, so that, we actually might be, and that's like my hypothesis basically, is we might be thinking this cancer protein is actually interacting with another one. So as opposed to trying to target it itself, you maybe can target the complex that they're forming, either by disrupting that if it needs this interaction, as
To
opposed
become cancer?
yeah.
Can you talk about when they are working together and you can then target it? What do you target it with?
So I'm not there yet, but in, I guess,
What's the whole?
hypothetical,
Mhm.
situation would be to design perhaps like an inhibitor. That's where actually having Barry as a collaborator comes in, because that's more his focus than like his expertise versus Alice's expertise. Or
And is this like antibodies that you're thinking, or are you thinking other kinds of things?
not necessarily antibodies, but what we call like just little small molecule inhibitors.
Mhm.
So they would actually be even smaller than an antibody.
Mhm, okay.
Is what's been out for other genes. Yeah.
And, we're all taught that cancer comes in many, many, many different forms. I don't know how many.
Exactly, and that's
So, you're working in general, or you're working on a particular type of cancer first?
So we actually, we look at lung cancer in the lab that I'm in, but the gene that I'm looking at, or the protein, if you will, is mutated in not just lung cancer, but actually like 95% of pancreatic cancers are caused by this mutations in this gene. So even though we look at the, I use like lung cancer cells and stuff like that, but it definitely could have implications for other types of cancer that have that mutation.
But at this point, the mutation that you're working with has been identified for sure in lung and in 95% of
Yep.
pancreatic.
Mm-hmm.
But not all lung cancers, just some.
Yeah, so it's actually 30% of
30%.
non-small cell lung cancers.
Okay.
Mm.
And what's the mutation?
KRAS.
Okay. Oh, okay. All right.
Sounds like a cereal.
I'm glad you know.
I don't know what it is. Unfortunately, though, we all know people have cancer, so we know this is very serious.
We know this is important, yeah.
Very serious.
And the whole idea, all I know is that the idea of being able to target it with it is groundbreaking, because rather
Yeah,
than giving everybody the same cocktail.
Exactly. So for some of the other genes where they have these actually established the targeted therapeutics, it's very remarkable for lung cancer to see, like, in a matter of months, what they can do.
So it's happening in some lung cancer already?
So yeah, there's, for example, there's some lung cancers that, not KRAS, but like EGFR, for example, that counts for 10% of lung cancer. That has we've like established therapeutics for that mutation, but KRAS is actually downstream of, or like underneath, in terms of signaling. So those drugs actually don't work on patients with KRAS mutations.
And you said it was so significant for pancreatic, but you haven't
Mhm.
started working on that yet.
No, we, just basically just because of the models, I just work with like lung cancer cells, but that would definitely be something, to
You'd be
extend in the,
interested in.
yeah,
Mhm.
definitely.
Wow.
Thank you for all the work you do.
Yeah. And it's amazing too, is you say you're really just in your first year of the actual research.
Well, because I've been in Alice's lab since 2017,
Okay.
April, but
Uh-huh.
we still take classes during then, so you're kind of,
Yeah.
so I finished my classes now, so I'm just like working on my project and stuff.
How much longer then do you think you'll have to complete your PhD? Is that a tough, is that an unfair
no,
question?
It's like one of those, I mean, it's like a joke where it's like, asking
Oh,
a woman
sorry.
her age,
Oh,
like when
okay,
you're gonna
yeah.
graduate,
Never mind, you don't have to.
No, I mean, it's like maybe,
But yeah.
it's like maybe 5 years, maybe 6, or
Yeah, so
like
you
total,
get, so people think it's, we know that it's never can be done in 3, and it certainly couldn't be done since you've done so much classwork in
Right,
3
right.
years.
yeah,
So that's why they say it's 3 to 5 years, but even sometimes longer,
yeah,
depending on
yeah.
probably how
Yeah.
complicated your research is. And
Yeah, I think
all
our
of that.
program's like 5.5 years
Yeah.
average,
Yeah.
so some 5, some 6.
Where would you like to see your research in 10 years?
Like on this project in particular?
Well, I don't know
or-
any other one, so yes.
Oh, I guess, well,
So what's the long-range visionary goal for this research?
well, assuming my hypothesis is correct, then I would really like to really start going more into like protein engineering and to really like design inhibitors, designing actual therapeutics, and testing them out and stuff like that. So I'd actually want to move more into like, I guess, design space of designing drugs.
And is that something that is happening in perhaps other, not your specific area of study, but the Institute for Protein Design?
Yeah, actually, I have Hannah Revola Baker, so
Mhm.
David Baker's wife's on my committee, so she actually has suggested that I go over there and, kind of start interfacing with them. So it's definitely something I want to.
Interesting, yeah.
Right, David
Wow.
Penn.
And what was your motivation to get into this field?
Really the idea of precision medicine, I guess the idea of, not just treating all patients the same and whether that's lung cancer or I really think it could be extended to not just cancers, but like maybe cardiovascular disease or just different kinds of disease settings, I think. Not just treating all patients the same, but really trying to understand what's driving it and maybe stratify the patients, I think is really why I got into it. I didn't know there was research kind of being done like this, and so I was like, oh, wow.
So your original bio says that you plan to continue stem cell research while focusing on cellular metabolism.
Mm-hmm.
Is that a true statement of the research you're doing now?
So we're not working with IP like stem cells so much, but actually I'm actually kind of getting a little bit more into metabolism and maybe thinking that there might be a role with that in terms of my project. So it's definitely something I'm still interested in. That's also why I have panel on my committee. So I feel like, so yes, I guess it's something I'm still interested and when I wrote that, I actually had Hannah's lab more in my mind. And so I did rotate in her lab, but I wanted to kind of extend, a little bit further. So I still actually want her, or have her on my committee. And so I do, I'm still actually very interested in metabolism and as well.
Are you still working with BioQuest? Does that exist every year?
Yeah, so they have it every year, but unfortunately they only let us do it our first year. So it is, so we do have outreach programs at the Hutch, kind of like Hutch High they have, and one where they bring high school teachers. So it's something that I actually want to do, even though I've kind of like fulfilled my technical requirements to do it, but something I'd like to do maybe next summer or something. Yeah, I'm actually gonna have, well, maybe have a undergrad again this summer, so.
That shadows you?
Yeah, so I've had one before, so
Oh, that's neat.
yeah, so it's nice.
How's the food and outdoor culture of the Pacific Northwest, which was your last line?
No, I'm actually I've actually gotten some hiking boots so I can do proper hiking in the rain out here. So it's actually, I've been on some that are quite peaceful. Like I was like, oh, this is quite pleasant. I don't know.
At least it's close.
Yeah. So yeah.
So it's okay being in Pacific Northwest.
Yes.
It's a little different than Southern California.
A little different. Yeah.
Although this year they've had as much rain or more.
Yeah.